Hello, grade 11s. I hope that you are still doing well and you are adapting to all of these new changes. There are going to be more changes coming in terms of how we communicate with you. We will be giving you more information about that next week, but we are going to be doing video conferencing eventually, which means we can kind of just all talk together. And instead of me just like showing you a screen and talking in the background, you'll actually be able to communicate with me in a better fashion, almost like we're in class again. All right, so let's just do a quick recap from last time. So we were talking about how cells signal each other. There are 4 different types. The ones that we focused on mainly was contact dependent last time. So contact dependent signaling means that the cells will be sending signals to each other and they're actually, like it says, in contact. So in these 2 cases here, they literally are attached to each other. So that makes it very easy for them to communicate back and forth. The other way also would be that they actually have receptors that will attach to each other, and that way they can also communicate. So that's called cell-to-cell recognition. Now, I gave you a couple of examples, mainly in the immune system. I'm not going to re-explain this slide, but just kind of give you the gist of it quickly. Notice here that these 2 cells are communicating by actually attaching. Okay, so that would be direct contact. And in this case here, the helper T cell would be the target cell because it's the one that's going to be sending out messengers to the other cells to make them perform certain tasks. This was the example of the CD8 cell. Notice, same idea. This is the antigen. So that could be a bacteria or a fungus, for example, that gets into your system somehow. It would then attach here, and then it sends out an enzyme that will destroy the cell. Okay, this was the other example we used were the natural killer cells. Natural killer cells normally are floating around in your blood. They will bind to healthy cells, and as long as they have this MHC marker, they will leave the cell alone. If a cell doesn't have that MHC marker, and that's usually if it's been infected with a virus, what will happen is it will then attach and it will realize that there's something wrong and it will destroy the cell. Okay. Now, I have created another PowerPoint. This is part 2. There will be 3 parts to this discussion. So if you want to follow along with this now, you can. So this is shared with you in OneNote. If you're having trouble getting things from OneNote, because I know some of you before didn't love it, just let me know and I can always just attach it to the email instead. Okay, so what we're going to do now is focus on the next 3, which were not direct contact. Now, what they all have in common, first of all, is that they are going to have a receptor on the surface of the cell, but they're also going to have something called a ligand. Now, what is a ligand? A ligand is defined as some sort of messenger that is going to be targeting a certain type of receptor on the cell surface and causing an intracellular response. Now, what could it be? It actually can be lots of different things. It could be a protein. It could be a neurotransmitter, which allows messages to be transmitted between neurons. It could be a hormone. It could even be something like drugs, like cocaine. Cocaine reacts this way. It could be some sort of bacterial toxin or even a virus. So it can be anything. It doesn't necessarily have to be something positive, but anytime it's something that is attaching to a receptor and it's causing some sort of response inside the cell. Okay, so this is just a little animation. I tried to find some animations this time. Now, as you can see here, what is the ligand? What would be that red thing? Now, when it attaches to the receptor, you can see there, it's very specific, notice in shape, which means it's not just going to be anything that's attaching to these receptors. Something is going to happen within the cell. All right, so let's look at specific examples now of this occurring. So if you take a look, now this would be considered local signaling because it's very local. As you can see, it's actually going to be triggering itself. So here, the little red dots here would be your ligands, and they would attach to the little purple part here, which is your receptor, and then it's going to elicit some sort of response within this cell. So that's called autocrine. An example of this that I found, I had to research it a little bit. You're not going to really have to know this, but it's always good just to have some background information. The immune system does this as well. It also, for example, if you were to get injured in some way, the immune system's response is to release histamine into the system. So that would be an example of mast cells releasing histamine during a immune system response. But again, don't worry, you don't have to really know that. I'm just telling you for interest. Okay, the second type is called paracrine. Now, paracrine differs from autocrine. How? They are very similar. They both have the ligand attaching to the receptor, but this time instead of actually stimulating or triggering itself, it's going to be signaling another cell close by. So it is still considered local in terms of its signaling. All right, I am going to give you some specific examples for this one. Now, what is going on in this diagram? Well, this is supposed to show you the synaptic response. Now, what does that mean? Well, we are looking here at two neurons. So this is a neuron, and this is a neuron. Now, in between two neurons, there's actually a gap, and that's called a synapse. So the question is, as you can see, let's just wait till it comes back again. All right, that little red thing that's going to come along, that thing there is actually supposed to be a message being sent along the neuron. So for example, let's just say I took a piece of chalk and pretend we're in class, and I threw it at Darshan. Now, Darshan would see it coming and react quickly and catch the piece of chalk. So that would be an example of what that red thing is. So it's a reaction to some sort of stimulus in the environment. So notice what happens when it actually comes. So see these little vesicles here. Now these vesicles here contain a neurotransmitter. And when the neurotransmitter, so when the message ends up at the end of the neurotransmitter will go and it will attach, and it's releasing the neurotransmitter. Now notice that the neurotransmitter is attaching to the receptors that are on the other neuron, the next neuron's surface. And that's going to allow the message then to cross over. Notice that it's localized. Here's a little bit slower of a diagram. Well, it's not moving at all, so that you can see that. So what happens here? So inside of these vesicles, for example, would be acetylcholine. And acetylcholine is a neurotransmitter. So when the message comes along, it's going to trigger the acetylcholine to go, and it's going to attach to the end of the neuron. They release inside. Notice that they are binding to these purple receptors on the other side. Now, can you see what it's triggering here? What is it actually letting into the next neuron? It's the sodium. So sodium is out here, it can't get in unless the neurotransmitter allows these receptors to open. So this would be an example of paracrine because they're very close in distance from each other. Okay, my last example here is the endocrine, which is what we're really going to be focusing on more in our homeostasis unit. So this time, this would actually be considered long distance signaling, right? Because the cells are no longer necessarily right beside each other. So what happens is the ligand, which in this case here would be a hormone, enters into the bloodstream, flows through. Eventually it will find its target cell, the receptor. It will attach to the receptor, and then it will cause some sort of response to take place. So if you take a look here, this is just an animation that I found online. You can see that the little shapes here are supposed to represent the hormones. Notice the specificity of them when they attach to the receptor. They're flowing from longer distances through the bloodstream, and they're going to target different cells and elicit some sort of response. All right, now specific example would be one that we've already talked about before. Now this one isn't like complete. It doesn't go into all the details that are happening within the cell, but I want you to just to get the idea of it. So the insulin here would be your ligand. It's attaching to the receptor, and notice it's eliciting a response by the cell by opening the proteins to allow the glucose to enter into the cell. All right, that's it for today. Please, what we'll actually do next time is we're going to talk about what actually happens when you are inside the cell. So we've kind of talked about how they're at this point signaled, but what actually is going to then happen within the cell. So what I would like you guys just to continue doing for now is work on your lab design. I would like you to send me your introduction on Monday just so I can make sure that you're on the right track. And yeah, have a good weekend, and we will touch base next week. Take care.